Welcome, everyone. Thank you for joining us for today's track on Forensic Science, a multidisciplinary approach. It is my pleasure to introduce to you Dr. Linda Birnbaum. Dr. Birnbaum is the Director of the National Institute of Environmental Health Sciences and the National Institutes of Health and the National Toxicology Program. She is a board-certified toxicologist and has served as a federal scientist for nearly 39 years. Prior to her current appointment, she spent 19 years at the U.S. Environmental Protection Agency. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Birnbaum's keynote presentation is titled Health Impacts of Environment Chemical Exposures. My name is Christy Jewell, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know you're experiencing a problem. This presentation is educational and it thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of your presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Linda Birnbaum. I will now turn the presentation over to her.
Hello, everyone. My name is Linda Birnbaum, and I'm Director of the National Institute of Environmental Health Sciences and the National Toxicology Program. I'm here to tell you a little bit today about the health impacts of environmental chemical exposures. First, just in case some of you are not familiar with what is NIH, it is the largest and the primary federal agency for conducting and supporting biomedical research in the United States and actually around the world. Our budget in 2019 is more than $39 billion, which is invested annually. And we are funding well over 300,000 researchers around the country, in every state, and around the world. Now, NIEHS, which is the institute of which I'm the director, is one of 27 National Institutes of Health institutes. And we are the only one which is located in Research Triangle Park, North Carolina, instead of in Bethesda, Maryland. We support a wide variety of programs supporting our mission, just like any NIH institute. We have our two vibrant intramural laboratory programs, one focused on basic biomedical research, and the other looking at toxicology issues, which is actually a cross-departmental program which involves CDC and FDA as well. Like any NIH institute, we have a clinical research program on site, and most of our money goes out to fund grants around the world. What makes this unique, in addition to our location, is that our focus is very much on disease prevention and public health, because if we understand what it is in our environment that causes disease, hopefully we can do something about it. So when we talk about our health, people often assume or go right away to the idea that everything in our health is determined by our genes. But I think we all know that it's not that simple, and that, in fact, our health is a combination of our genes and our environment. And here at NIEHS, we define our environment very broadly to include not only synthetic chemicals that might be used, for example, in agriculture or in our consumer products, or things that are emitted by industry of their tailpipes and go into our air or our water, but also things like the medicines that we take, the food that we eat, the stress that we all live under, and, for example, many personal care products. All of these things can have impacts upon our health. A realization that's hit us very strongly within the past couple of years is that a good start lasts a lifetime, and unfortunately, so does a bad start. And we're understanding that things that affect our development very early in life, including before conception, but certainly in the womb, during infancy and childhood, in our reproductive years, and so on, can have impacts on our health later on. And this is a whole new field called the developmental origins of health and disease. We're also understanding that when we talk toxicology, which is really the science of safety, is that much of the idea that how much you are exposed to or your dose makes the poison comes from a statement that Paracelsus made about 500 years ago. We know today that it's more than just the dose. It's also the timing and the acute versus chronic nature and where you are in your life stage that can make a difference. But the dose does play a relationship to what the effects that we're going to see. Now, when you're dealing with forensic toxicology, you're often dealing with acute poisonings, and you're dealing sometimes with lethal effects. But when we're dealing with impacts of environmental exposure on our health, we're often focusing on things that happen at low doses and often exposures that occur repeatedly or in a chronic measure. So as I said a minute ago, dose is not the only concern, but factors such as our genes, which help determine our susceptibility, whether we're male or female, when in our life it occurs, and the microbiome plays a role in susceptibility. So when we talk about forensic toxicology and environmental health, how that kind of differs from what we're dealing with environmental health research, forensic toxicology is focused on helping the medical and the legal professions deal with certain issues. But environmental health, we're really trying to understand how the environment affects our health to inform disease prevention. But there are many things that are similar between the two fields. First, in both cases, we're going to be collecting, preparing, and storing tests and various biosamples, and we want to store them so that they're stable. We have to all think about the dose and when the timing of the exposures and where the exposure comes from. Both in forensic toxicology and environmental health, we have to think about how long the chemical hangs around in our bodies, whether repeated exposure is going to build up a dose in our body, the bioaccumulation issue, and whether the chemical to which we're exposed in the environment is actually transformed when it gets into our body into something else. But while we have certain differences, we are also looking at similar ways of screening and measuring some of the exposures and some of the early responses to exposures. So we may be using GC mass spec or high resolution mass spec or NMR, for example. And for bioresponses, we may be looking at cytokines, we may be running various amino acids, immunoassays. There are different things that we want to do. What's become absolutely clear is that when we're studying public health, it is absolutely essential that we integrate the tools and the results we find from epidemiology data with those that we find from toxicology data, which in terms of experimental toxicology is usually dealing with animals or in vitro or in silico or computational kinds of data, and that we absolutely need these interdisciplinary collaborations in order to understand the impact of our environment on health. So I'm going to give a number of examples. These are in no way complete, but they're hopefully to just give you a taste of some of the things that we are interested in when we're studying environmental health and the impact on people. So first I'm going to go through a little bit about water, then I'm going to talk about air, then I'm going to talk about food, and I'm going to talk about soil. So when we talk about what's in our water, there are lots of things in our water, and things that we can know. There can be pesticides, there can be different kinds of bugs or viruses. We can have things like plastics. We can have the chemicals known as PFAS, which I'll talk about later. We can have things like lead. I think everyone is familiar of the tragic situation in Flint, Michigan of a couple of years ago. And we can have many other compounds in our water, such as mercury. But this is, again, is just a tidbit or a tapping the surface of what might be in our water. And I'm going to give you some examples. And I'm going to start off with arsenic poisoning, because drinking arsenic contaminated with drinking water is a major problem in several parts of the world, but is actually a problem even where I come from in North Carolina. And arsenic, some of you may be familiar with the play Arsenic and Old Lace, in which these lovely old ladies are busy putting gentlemen callers out of their misery by giving them high levels of arsenic. But the poisoning by arsenic is not limited to tales, stories and tales. It has also occurred in real life. And unfortunately, again, here in North Carolina, we've had several recent incidences of both murder and poisoning by family members. Blanche Moore actually dispatched of, I believe it was 4 husbands, and 1 boyfriend survived her attempts, and she was caught. But there was another tragic incident here where a young woman poisoned her husband with arsenic, and he did not survive. So very high levels of arsenic can lead to fatalities. But we know that at lower levels of effects, there are many different things that arsenic can cause. Arsenic is a naturally occurring element, and it can occur both in organic and inorganic forms. And there's a whole variety of health effects. For example, at high levels of exposure, it's associated with cancer in the skin, the bladder, the lung, also the kidney, probably the liver. There are effects on the brain, both developmental and in adults. There's effects on the immune system. There are effects on our respiratory system. There are effects on our hormonal systems. Arsenic can act like an exogenous estrogen. And as I've already mentioned, high doses can be associated with death. Now, in a study that we've been carrying on for almost 20 years, or I should say our grantees from Columbia University have been studying a cohort of 35,000 people in Bangladesh. This is an example of good attentions gone awry, where WHO in the 1970s put in pumps to get to groundwater, which was uncontaminated by microbes. Unfortunately, and unknown to WHO at the time, this groundwater was heavily contaminated with naturally occurring arsenic. And it took over 10 years before people began to report examples of arsenic toxicity. But following people now for the last 20 years, we've been able to observe some of the adverse effects in this population of increase in mortality, problems in cognition and motor function in children, effects on the heart and the lungs, and effects on skin lesions, which you saw in the last picture in the last slide. Now, the effects that are occurring in Bangladesh at very high concentrations have also been observed in Maine, where our grantees have looked at the IQ of children who have spent essentially their entire life in Maine, in most cases on well water, and we know that there's naturally occurring arsenic in the water in Maine. And what they were able to show that children with higher arsenic exposure showed a reduction in IQ points. Now, this is not, you can't pick this up on an individual basis, but you can pick it up in a population of children and find that those drinking higher arsenic levels, and this was only the levels that they were drinking, was only 5 parts per billion, which is within the maximum contaminant level that has been set by US EPA. The level that EPA has set is 10 parts per billion. So this is really increasing our level of concern for arsenic at low levels of exposure. We're also finding associations in a variety of epidemiology studies, and there's some animal data to provide biological plausibility that there's an association between high arsenic exposure and increased risk of type 2 diabetes. This is especially true of individuals who are overweight or actually obese. Overweight is usually considered a BMI greater than 25 and obesity greater than 30. And the findings really support a synergism ongoing between the risk of type 2 diabetes. I think most of us know that overweight is a risk factor, but exposure to arsenic increases that risk as well. Now, we're also finding that low levels of arsenic are associated with immune suppression, and this can be picked up in response to bacteria. This is Pseudomonas aeruginosa, which can be a bacteria that can colonize the lung. and this suppresses our immune responses monitored by the secretion of cytokines. And if we look at MMA, which is one organic form of arsenic, and actually we're understanding that MMA can be the most toxic form of arsenic, what we can see is a complete difference with a lot of more cytokines produced by the as a result of bacterial infection, but then we actually get a decrease in that with the exposure to MMA at very low doses. Now, I mentioned that we can see effects related to early life exposure, and unfortunately, there's a classic study of this that happened in northern Chile, up in the Atacama Desert. Antofagasta is a fairly large city in which people were exposed to high levels of arsenic in their drinking water from 1958 to 1970, and this was because there are many copper mines in that area, and they rerouted the river to run through the copper mines to wash out debris from the mines. But then 50 miles downstream, it was the drinking water supply for this city. And unfortunately, what we found is that for people who were exposed during this time, you can see that their arsenic concentration in these people changed. Well, this is actually concentration in the water, increased dramatically because of the water source. And we did find that people who were exposed early in life to arsenic, especially in utero or in childhood, had an increased risk of several kinds of cancer, type 2 diabetes, and hypertension. So this is actually showing you that it took 40 years to have the 40 years of exposure. Okay, I'm sorry. Arsenic exposure started to increase in around 1958, and it peaked in about 1970. But you can see that lung cancer, which is associated with this increase in arsenic, has a very long mortality latency before you find the disease, and it was at about 40 years. So what we can find is that the time of high exposure, you have almost a 40-year risk before you see the increase in cancer in the population. What we're seeing in our human studies, again, we have biological plausibility that support this from our animal studies. If mice are exposed starting in utero, so before birth, as well as throughout their life, to arsenic in the drinking water, what you find is at a dose of 50 parts per billion, and remember I mentioned that the regulatory limit in the US is 10 parts per billion, we are finding a very significant increase in lung tumors in the mice. And this is true both in males and female, but we actually see benign tumors, things like adenomas as opposed to carcinomas, occurring at the lower concentration in females as well. And an interesting thing here is at the very high concentration of 5,000 parts per billion, we do not see a significant increase in cancer. And part of that is because the mice have other problems that they may die before they develop the tumors. So that was just giving you an example of something in water which can cause a problem. I want to go over some of the things that are in the air we breathe that are problematic for our health. And these are a variety of things. I think people are familiar with the regulatory levels or that we might have on ozone or on, for example, particulate matter. But in addition, we can have biologics in our air, things like mold. We have smoke, and I think people are familiar with the secondhand smoke issue. We can have volatile organic chemicals, things like formaldehyde, and we can have gases as well, things like sulfur dioxide and nitrous oxide as well. And of course, things like asbestos and allergens can also be present in our air. So the answer I'm going to focus on are some outdoor and indoor air exposures. Our outdoor exposures are what we regulate. We do not regulate our indoor air exposures, which may be problematic. But in our outdoor air, we can be exposed to, obviously, vehicle emissions and industrial emissions are things we're always concerned with. Ozone is generated when we have generates smog, I should say. We have particulate matter. We have polycyclic aromatic hydrocarbons, known human carcinogens. And we can have things like dioxins as well. And outdoors, we have many similar products that we have indoors, like, for example, particulate matter and PAHs and so on. But we also have things like mold and dust, We have the vapors that we get from cleaning products. We have flame retardants and a whole variety of different kind of chemicals indoors. So when we talk about air pollution and health, we know that approximately 8 million people a year worldwide actually die from air pollution. And while the majority of those are in low and middle income countries, it is also a problem in our country as well. Of the deaths from pollution, it's approximately half and half, that about 4.2 million are related to outdoor air pollution and close to 4 million are associated with indoor air pollution, especially related to cook stoves using biofuels as their fuel source. I think we need to realize that while most air pollution deaths are related to non-communicable chronic diseases such as cancer and stroke and heart disease and other kinds of lung disease, never mind type 2 diabetes and obesity, but I think it's important that morbidity worldwide is also related to more related to non-communicable rather than infectious disease today. So some of you may have heard about the London smog epidemic, which occurred, or episode which occurred, my goodness, over 65 years ago. There have been similar episodes that have occurred in Belgium and in Donora, Pennsylvania area and in other parts of the world. But what happened was the London smog, there was an air inversion at the time in the wintertime, and there was an air inversion, and much of their heating and electricity generation was being done by coal at the time. And as many as 4,000 people died during this air episode. Many died from influenza, which again increased in part because of the suppression of the immune system, like I mentioned with arsenic before, but other reasons also. Many people died, they didn't know why. And what you see here in the light blue bar is that when the government estimate following in that winter related to the smog was a tremendous increase in the amount of mortality. Now, I can say this is not unique to London. I mentioned several other episodes. Every time there is really bad air pollution in a city or a dense area, we see an increase in hospital visits and deaths, which are associated with the heavy air pollution. So we're also finding that it's not only acute mortality, but we're also finding that there are effects on the developing fetus, so that we can see there's a clear association between particulate matter exposure during pregnancy and decreased birth weights. And you can see this both when we're looking, for example, things like elemental carbon and sulfur, or if we're looking at some of the different metals that have this effect. And this is more pronounced when exposure is later in pregnancy. And some of our components, for example, something like vanadium, has a very dramatic effect on the weight of the baby. Now, we also know that asthma increases, both the induction of asthma and then exacerbations. In other words, people will have more asthma attacks that are associated with air pollution. And we've known for a while and have more and more data that show that you live close to a major roadway, within 200 meters, there's an increased incidence and there's a risk of increased incidence and persistence of asthma. And that this occurs not just during early life exposure, such as childhood, but can also occur in adult life. So actually this Bawadi study is following a longitudinal cohort of 45 people in Tasmania that they followed for 8 years, and showed that you had people becoming asthmatic in that time frame associated with the greater exposure to PM 2.5 and to black carbon as well. And this is on your left on your screen is what's happening in children, and that there is also the association that we are seeing in adults as well. So what else is happening to children in addition being more at risk for low birth weight, being more at risk for asthma. There's also effects on learning, memory, and behavior in children. And this is a study just showing the association with an increased risk at the higher levels of air pollution exposure. Adducts are what are formed by PAHs. They actually bind to the DNA and can be measured, for example, in the cord blood of the mothers, so that we know that this is associated with in utero exposure. And you can see with higher levels of adduct formation, there is a greater risk of attention deficit hyperactivity disorder. We also know that with exposure to other kinds of air pollution, not only the PAHs are present on the particulate matter, but with nitrous oxides, we see impacts on learning and memory as well. And one thing I want to point out here, may come back to later, is that we often see sex-specific or sex-dependent effects. Sometimes males are more sensitive, sometimes females are more sensitive. So I often say that's just vive la différence. But I think, again, we're also seeing associations of this traffic-related air pollution being associated with increased risk of depression and anxiety. So effects both on learning and memory and on behavioral endpoints. Now, this is just some concerning data. I think many of you are aware of the dramatic increase in children on the autism spectrum, or I should say people on the autism spectrum, which has increased from about 1 in 120 to 1 in 59 at the last CDC studies. But what you can see here, depending upon the different measures that are used, in almost all cases, exposure to, and again, this is traffic-related pollution, is associated with an increased risk of a child being on the autism spectrum. And this is related to maternal and very early neonatal exposure. And there are many more studies looking at air pollution and finding this relationship. But something that I wanted to point out is that we see that this is consistent in multiple studies, although within a given study, there may be some differences. Now, it's not only early in life and age that particulate matter is a problem, but we know that there is growing evidence for particulate matter being associated with neurodegenerative diseases as well, both for all kinds of dementia, for Alzheimer's and Parkinson's disease. So in this study, they looked at almost 10 million subjects from 50 US cities, and this is the data for the 50 cities. And what you can see, if you put all the data together, there's a clear statistically significant increased risk of Alzheimer's disease as the particulate matter levels increase. And this is just some data with Parkinson's disease, and this is looking, in this case, at the nitrous oxides. And what you can see if you look at the curve is the increased risk, higher odds ratio of developing Parkinson's disease in association to traffic-related air pollution. Now, I mentioned that I have a lot of concerns with household air pollution, which is largely, much of it is related to cooking with open fires and biomass stoves. It could be coal, it could be dung, could be wood, but also kerosene. And the estimates, before I had given you an estimate of 3.8 million, WHO recently estimated 4.3 million deaths associated with household air pollution. And these associations we find in the women, an increased blood pressure related to their carbon monoxide exposure getting out of this. There's also lots of data associated with the particulate matter. I think when we're talking about this indoor cook stove exposure, women and children are disproportionately exposed. Children have a higher risk of pneumonia related to this exposure, and women are at increased risk, for example, of lung cancer. In addition, even in our country, there are studies coming out of the NIEHS sister study showing that indoor wood burning, even once a week, is associated with a higher risk of breast cancer for women in the US. And we do have many of our rural and tribal populations are doing quite a bit of wood burning, and I think this raises some concern. We also understand that I've talked about air pollution from mainly particulates and some gases, but things like gasoline vapors can also be acutely fatal. And this is just some studies showing that a man went into a control room with the transporting unleaded fuel, and he actually died from the exposure that he had to a variety of things from the vapors. I think at least in the past, levels of benzene in your gasoline could be as high as 5%. It's lower now. But this just shows some of the data of the levels that were in the person who died from exposure to, in this control room, to unleaded fuel, the vapors of it. And they believe that this person died very, very rapidly. Now, benzene, I think people are well aware that this is a dangerous chemical, but we do form it naturally, just hopefully not very much. It is used in closed processes in many industries, but it can also escape into the air, and where it can be used as a solvent in a variety of industries. The major route of exposure is inhalation, although there can be some ingestion if benzene is in your water or in some of your food. Benzene is a known human carcinogen. We've had evidence for that for close to 100 years now. And in addition to causing especially different kinds of blood cancers, it is also associated with effects on our cardiovascular system, our neurological system, and again, effects on the immune system. So looking at some of this data, what we can see is the increased risk of all leukemias associated with benzene exposure. And this is whether it's acute lymphoblastic or acute myeloid leukemia. I think one thing, if you look at the dose response curves here, what you can see is the greatest effect is happening at the lowest levels of exposure, where you have a very steep dose response curve. We know that there's a higher risk for childhood leukemia when the parents are exposed to benzene. And as I mentioned, related to both smokers and non-smokers, there's an increased risk of heart disease. And we did find an increased risk of neurological symptoms of residents who were exposed or who were exposed in the Gulf states to, for example, the Gulf oil spill of 2010. So switching away from air and water to what's in our food, again, unfortunately, sometimes we have bacterial or viral contamination. We have all kinds of different contaminants that can get into our food, either because of the growing of the food, the conditions under which it's grown, or how the plants or the animals are watered, for example, or sometimes related to transfer from containers in which food is stored. So as I just said, our food can be contaminated by our natural environment. Arsenic, for example, is a problem or can be a problem if rice is grown in areas where arsenic is present. We can have, for example, microbes from our natural environment, pesticides or PCBs, but I also mentioned food packaging. And so things like BPA, which has been removed from baby bottles and sippy cups, but BPA and its substitutes are still present in other food contact material, phthalates, and the per and polyfluorinated alkyl substances, which are of growing concern in our country and around the world. There's a whole variety of effects that can be associated with exposure to our food contaminants, and again, varies depending on the contaminant you're talking about it and the inherent susceptibility of who's exposed. So I mentioned the PFAS substances, and I think our concern has grown for these because they are produced in very, very high amounts. They have now led to contamination worldwide, including the Arctic, where they were never used. There are thousands of them, with more being developed and produced all the time. They move through the environment, and basically they're known as forever chemicals. They will never leave the environment. The carbon-fluorine bond is probably one of the strongest bonds that exists in nature. We're using them, or they've been developed, because they provide resistance to grease and water and oil. So they've been used to make our frying pans and our raincoats and our hiking boots and many other important uses. But they're also being used in many food contact materials. And there are alternatives. The top two, the PFOS and the PFOA, have actually, their production has stopped in the United States. But their alternatives, for example, like this Gen X, There has been very little information available on it. I will say that as we study some of the alternatives, if they are a PFAS member, we're finding that they do many of the same things that PFOS and PFOA do. So PFAS health effects, that has again the laundry list of health effects, including endocrine disruptions, specifically the thyroid here. We also see the increased risk associated with PFOS and increased risk of diabetes. And with PFNA, which is similar to the PFOA I mentioned before that had 8 carbons, this has 9 carbons. And what we're seeing here, again, is an impact on executive functioning, that's basically learning and memory, in children related to their early life exposure. Again, it's early life that is part of the issue. I mentioned that food packaging materials can contain PFAS, and a study was done 2 years ago, which showed that almost half of food contact papers and many paperboard samples contain PFAS. That was actually validated by going into a subset of the hundreds of samples that they looked at to see what kinds of PFAS they were. And there were many that have not previously been reported. But while a lot of the focus on PFAS has focused on what's in our water, we are now understanding that what's in our food may be a major contributor to what's in our bodies. The other compound or group of compounds I wanted to mention in our food are the phthalates. And this is a huge group of chemicals that are often used to make plastics more soft and flexible. Many of you may remember rubber ducky. That used to have certain kinds of phthalates in them. Those phthalates are no longer being used, but we have alternatives. Alternative phthalates, and again, like the PFAS story, we are finding that the alternatives may be problematic as well. Now, I mentioned the food contact materials. DEHP, diethyl hexylphthalate, is one of the main phthalates that is present and contaminating our food from those sources. And again, not only do they come from the packaging, but also the processing and handling, say, in the factories. Again, a long list of health effects, including effects on our endocrine system, and including effects, for example, here, increased risk of developing ADHD related to in utero diethyl hexylphthalate exposure. We're also finding that prenatal exposure is associated with increased risk of overweight. So again, DEHP, increased risk of increased BMI, so do some of the other related phthalates. And one of the things that has been shown repeatedly in animal studies and in human studies, that exposure to phthalates can act in an anti-androgenic member. We see effects on the developing male reproductive system, which is maintained into adulthood. And we also see that the sperm produced in this case are less mobile, which is a problem related to fertility. Where are we getting phthalates? Again, often in food packaging, but where do we find this to be very problematic, especially related to fast food intake? So, for example, what you can see here is especially, for example, children. Again, because children eat more per their size than adults do, we can see that intake of phthalates is associated with higher levels than in adults and adolescents. But what we can see here also is the increase levels with no phthalates in adults and adolescents, middle levels of phthalates, and high levels, of fast food. And we infer from that, actually, and have measured the levels of a phthalate in their urine. So I mentioned that rubber ducky is no longer has the same phthalates it did years ago. For example, it used to have something like DEP, and that's been replaced by this DIBP. And you can see that when a regulation goes into effect, you see a dramatic decline in the levels of that phthalate in the urine of people who might be exposed. And you see an increase in the replacement phthalates in the urine of exposed people. Now, I did want to mention the kinds of exposures, environmental exposures we would have from our land, soil, and settlement. And again, you see some of the favorite actors coming back: arsenic, mercury, pesticides, dioxin, PAHs, all kinds of metals and so on. So there was a pesticide poisoning, and I'm trying to use some examples that I think are very relevant to forensic toxicologists. This was a young boy poisoned by sulfuryl fluoride, which is a pesticide. It was used to fumigate his home, and the child entered when the family was told it was safe. Unfortunately, this child was poisoned by the residual fluoride in his home, and he suffered severe brain damage, and obviously that became a case for the courts. But this is an example with a high dose exposure to this chemical was very problematic. Now, we also know that pesticide exposure, whether it's a herbicide, a fungicide, or insecticide, or a disinfectant, there are many sources of exposure that can be agricultural. And so if you're a farm worker, you can be exposed, or if you live near a farm. But some agricultural things, of course, remain on our food. It could be things that we use in our homes, or it could be occupational. And there are many health effects associated with exposure, including cancer, neurological effects, endocrine effects, and death, even at very high levels. So this was, I think, an important study looking at the effects of organophosphate pesticides, and this was namely chlorpyrifos. And what they saw here was there's children are, in fact, often they can actually be involved in some of the field work in Ecuador, but certainly their parents bring it home on their clothes. And if you look after Mother's Day, what you can see is an increased problems in children's behavior associated with the exposure that the tremendous floral production and that was used related to the production of large numbers of flowers for Mother's Day. And so we believe that there are effects on the children's neurobehavior. Now, I actually want to go back to this again because there's lots of evidence that this is exposure when the children are young children. We also know that exposure to organophosphate pesticides in utero is associated with lower IQ in the children. So I mentioned that air pollution is associated with increased risk of autism. We're also finding, again, the beauty of longitudinal epidemiology studies. The observational studies have so much power when they're done longitudinally. And in a study that was conducted in Finland, they were able to show that mothers who had the highest levels of DDE early in their pregnancy were more likely to have a child who developed autism. And DDE is the major metabolite that is produced from DDT exposure. Now, I mentioned the organophosphate exposure, and again, chlorpyrifos, and the IQ, but it's also related to decreased motor function. So what you can hear is that there are impacts, for example, with those who are most highly exposed to gross movement, fine movement, and composite movement as well. In this study, this was done in China, and they compared the motor functions of neonates with 9-month-old Chinese infants, and they found exposure not only to chlorpyrifos, but NIEHS, which some of you may remember is the pesticide, the organophosphate pesticide that was recommended for use for Zika spraying, was associated with impacts on motor function. This is some of the data, the animal data, which supports what's been observed in observational human studies, which that exposing rats during most of this is gestation day 7 to postnatal day 21, is associated, and with quite low doses actually, is associated with neuroinflammation in the brains of the neonates. And this is again associated with exposure, what's called the perinatal period, during gestation and throughout lactation. And the really interesting thing here is that the effects were sex specific. They were more pronounced in the males than the females. But the graph here shows you the very clear dose response for the evidence of increased inflammation and being associated with problems with working memory. Okay, let's see it. I wanted to switch here to talk a little more about inherent susceptibility difference. So this is just a picture of a group of my employees here at NIEHS. And if you look at it, you can tell that we're not all the same. And I think that's something that we always have to remember, that there's tremendous variability, not only in how we look, but in how we respond to exposures. So for example, we know that with arsenics, that there are genetic polymorphisms in genes related to the transport of arsenic into the cells, how arsenic is metabolized, how arsenic can produce oxidative stress, and how it damages DNA, which is dependent or can vary with certain different kinds of alleles of certain genes. We know that as far as the food that we eat, people handle, for example, folate and vitamin C. Some people can clear it or remove it from their bodies very quickly, others can't, and there can be tremendous distance. And we know, for example, with the microbiome, that while we used to only think of the bacteria in and on our bodies as trouble, we now realize that the microbiome, a healthy microbiome, is essential to our health. There's really a synergistic interaction going on, but the microbiome can play a role in our metabolism, for example, of arsenic. So some people, whether it's because of their genes or what they eat or their bacterial content, may handle, some will be more and some less sensitive to arsenic. Now, there's again a very interesting study done in northern Chile, in not in Antofagasta, but in neighboring regions, where they found a population of indigenous people who had a polymorphism in this AS3MT gene, which metabolizes arsenic so it can be removed from the body. And they found that these people were living in areas with very high arsenic in their drinking water, but did not have, were not showing evidence of lung or bladder cancer. And a lot of that was because they were getting rid of their arsenic very quickly. They found that the population, for example, or in this case, they looked at mestizos who were living in a nearby area, and they were they didn't have the altered gene, and they were susceptible. And then this has also been seen, for example, in a European population, and has also been seen in Bangladesh. Now, we also know that in Bangladesh, where I mentioned there's been this problem in many areas of Bangladesh with the tube wells, which led to arsenic contamination of the water people were drinking, we found that the number of copy numbers, variations in several genes, led to an increased higher risk of arsenic-induced skin lesions. And DNA copy number variations are being shown to be associated, many CNVs are being shown to be associated with a variety of diseases. I think the important thing is we don't see this effect in women, and it's only being observed in men. So that's sex specificity that I mentioned before. And the copy number variation is also present, for example, when we look at the increased risk of autism spectrum disorder and early life exposure to ozone. And if you look at this 4x4 table, what you can see is for those individuals who have a high number of copy number variations related to a gene related to autism, plus have high levels of exposure to ozone, those are the people at greatest risk of being on the autism spectrum. While as people who have low ozone and no copy or very limited copy number variation are essentially almost not at risk for autism spectrum. So I think, again, the specificity is important. We also know that benzene, that there are differential susceptibility here, and genes are involved in metabolizing, for example, benzene, and metabolism gets rid of the active benzene metabolite. And so there's evidence that for people who can get rid of benzene very quickly, they are going to be less susceptible to the adverse effects of benzene. Now, we're also finding, for example, that the levels of different contaminants may vary, for example, by race, but sometimes this may be related to food packaging. So these are looking at 4 different PFAS. This has C8, C9, and C10. And this one, instead of a carboxylic acid, has a sulfonic acid at the end. And what you can see is that people who are eating a lot of fast food from cardboard containers, which have been lined by PFAS contaminated material, in this case, they are more likely to have elevated PFAS in their blood than, in fact, if they were low consumers. And the hypothesis is that these African American subjects were just eating more fast food than, for example, the white subjects in the study. Now, diazinon is another kind of pesticide. And again, we're seeing more another evidence of sex specificity here. Well, this study was done in mice. And there was evidence here that diazinon altered the microbial community. This is another organophosphate in the gut of mice. And the changes were much more pronounced in the males, where you can see changes up to 2,300, from 50 to 2,300, as opposed to only 5-fold in the female mice. And again, this may explain why the male mice are more sensitive to the neurological effects of diazinon than are the females. Now, we also know there's a gene very important in trying to detoxify organophosphates, and this is a gene called PON1. And we're showing that people, that there's polymorphisms in this gene, and people who are polymorphic have more likely to suffer toxicity from organophosphate poisonings. In one of our longitudinal studies of a birth cohort, where we started measuring levels of exposure in the mothers while they were pregnant, and then continued to follow them and their children, we showed that PON1 does play a role in the individual susceptibility. And we're also showing that this may play a role in the behavioral effects that we see organophosphate can create. So we believe that by creating healthier environments, we are going to improve health. We're going to inform health protective policies and regulations. Our focus at NIEHS is on prevention. We want to inform intervention and remediation strategies that promote behavior change. We want to build environmental health literacy through outreach and education, because while we can't change our genes, at least not yet, even with the advances in CRISPR-Cas9, we can change our environment. And by, I mentioned one example before of regulations having an impact. But for example, when EPA changed its drinking water maximum contaminant level in 2006 from 50 micrograms per liter to 10, what you can see is a decrease in the levels of arsenic in urine in the general population following that reduction. And that has impacts because there's an estimated decrease in the number of cancer cases that could be associated with arsenic exposure in drinking water. Educational interventions can also work, where basically in Bangladesh, the children were taught in school what they needed to be conscious of, like the well handles that were bright red were ones they should not get water from. That led to families switching to lower arsenic in their waters, so they had lower arsenic in their urine as well. And again, those where there was switch to had better education did better. This is a really beautiful example of improving air quality, improves health. There are a number of examples here that we know that reductions in PM decreases mortality risk, especially from heart and lung problems. We know that decreases in nitrous oxide and PM lead to increases, better function. And that's actually this slide over here. As the PM concentration decreases, what you can see is that the measurements of lung function, the FEV1, get better. And again, this is following 3 cohorts of 11 to 15-year-old children in Southern California, living in different regions with different ethnic backgrounds. But as the air improved around LA, the children's lung function, the children who grew up with less air pollution had better lung function than the children who grew up with more highly polluted air. We can also work on education to reduce the pesticide take-home pathway. I mentioned that related to the children in Ecuador, whose parents were working in the flower industry. But we know that pesticides are higher in homes and vehicles of farm workers compared to non-farm workers. But we know that farm workers can reduce their exposure by wearing gloves and leaving their work clothes at work. before they return home. So in summary, the interaction of our genes and our environment determines our health. There's lots of role of individual variability, which influences our biological response. Forensic toxicologists and environmental health scientists, we're all trying to understand how chemicals can affect human health. Again, as an environmental health scientist and a toxicologist, I'm often focusing on low-dose chronic exposure, but by working together, both individually and through policy change, we can modify our environment to reduce harmful exposures and improve our health. So thank you all. And while I can't answer questions directly now, I'd be very happy to receive emails, and I'll be happy to answer them. So thank you all. I hope this was helpful.
Thank you, Dr. Birnbaum, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Dr. Birnbaum will be following up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.